they learn a lot of the actual techniques on site. So when they go into the BC Cancer Agency or a hospital, they learn just huge amounts of information. And they get much more motivated to learn it because they actually see where it's getting applied. And so my experience is they take sort of a number of master's level classes, often don't really understand why they're learning what they're learning, but the on-site experience is where they're getting just embedded into it and really enjoying that kind of learning. So I don't know if the same would happen the other way around. If we had medical students who take some computer science classes to become conversant, but then we put them with our students in computer science, where they really bootstrap what they've learned and get much further.
Great. Thank you. Question over here.
Well, maybe following up on that, I guess the big issue is what are they learning? And you know, you talked about when you were studying, you learned how to manage data that you knew was data. You knew how to apply the algorithm. You knew which was appropriate and what wasn't appropriate. You got this experience, this feeling. So given that you knew you had data, what they do with that data? And I mean, the same is true with medical students. You know, they have all this knowledge of physiology, biochemistry, and then they see patients. They know the limitation of that knowledge. So do you really think that I take it from your answer, you don't, that just getting medical students learning how to code in Python or computer scientists doing a few chart reviews or something like that may not necessarily do it. So could we flip it and focus not necessarily on the processes, but focuses on the errors that can emerge? And just everyone be humble and say that this is the best we can do.
I think that's exactly right. So what I see is that my students, because they know where the data is coming from, they have more of an intuition on what that data means, and they can ask more meaningful and deeper questions to the clinical scientists. They're not themselves clinical, they're not running those experiments, but they're working alongside clinicians, so they see the source, and that's where they're getting depth. Whereas my training was just assume that the data I got was correct. They can probe a little deeper into it.
But is there a room for someone, a third group, that are the skunk works, that they always assume everything's wrong, and they always destroy all these wonderful future scenarios? And what would that group be like?
Would that be since I'm looking at Dawn here, I'm just thinking we have statisticians and our people in the Institute for Clinical Evaluative Scientists. I think they really take that very critical stance and stand back. So maybe I think you're right. And Peter, it's nice to see you again. I feel like I'm on my PhD defense committee. Peter used to grill me, so I'm glad you took the answers. There's a question over here.
Yeah, I think
it's Kareem.
Yes, it's Kareem. Just to follow up on the question, I think one of the things you said toward the end of your presentation is, well, we can look at, diet patterns and see whether people are better on certain diets or whatever. And I think, we call that hypothesis testing.
Mm-hmm.
And it's easy for Google to do an A/B test on the millions of people who come to their website and randomly allocate to different web pages. But in medicine, we call that randomized controlled trials. And there's a huge cloud of ethical issues because people who presumably come to Google do it on their own free will, whereas those who come into hospitals and into our institutions do not. And so we don't have that ability to experiment on them. So it would be interesting to hear you say something about that.
Well, that's very good question. One of the interesting things about these wearables when they become ubiquitous is that many people are doing exactly that. They're themselves deciding which test they're part of. And you can then randomize the wearers to get the data you want. But I know there's deep ethical issues about what kinds of tests you can do on people, and I'm hoping ethicists have a better understanding of how to handle those. But I do think that medical devices are coming that are be able to just simplify a lot of this type of testing.
So maybe I'll take the last question, and then we'll hold the other questions over. So Mahat, please.
This is more of a technical question. You talked about the challenges of detecting false positives and actually finding signals. A lot of way epidemiologists kind of were setting up standards and sort of robust methodologies. Are there such robust methodologies or standards that you've thought of? And what are your thoughts around how you actually determine that a real signal exists?
Yeah, so that's one of the things that we found. First of all, at least in what we were doing, there weren't really good standards. It was the first time that these particular techniques had been used on FCM data. The big thing that, I guess for me, the eye-opener was how I felt almost our roles were being reversed. That as computer scientists, people expected us to want a lot more data. And so the medical people, the clinicians were saying, we can just generate way more data for you. And we were worrying about false positives. So we were saying, let's try to figure out which parameters have the highest correlation with the signal we're looking for, so that we can minimize the prevalence of false positives. It was a pretty new area for us, and like I said, we really saw that we're working from very different angles to try and get this data. We ended up looking at data with 65 million parameters, which is already pretty big. We limited the space enough. We brought the dimensionality down enough that most of the signals we saw seemed to be true, not false. But it definitely, the first round when we ran this stuff, we just saw all sorts of things which were extraneous. And I think we all learned that we need better protocols, exactly what you're saying, for this type of data. But it was such a new area that we hadn't gotten that far.
So thank you for your questions, and we will have time at the end of the morning session to dig a little bit deeper into some of them. So thank you, Professor.
Thank you.
Now it's my pleasure to introduce Steve Shearer, who is a professor in molecular genetics, the director of the Center for Applied Genomics, as well as the executive director of the McLaughlin Center. So his presentation is entitled "The Missing Open Science Project: 10,000 Autism Genomes in the Cloud." So welcome, Steve. Thank you.
Okay, thank you very much, Trevor. I'm also proud to say I'm a senior fellow here as of last fall, so maybe I'll make it on the list next year. So I'm going to take a little bit of a different approach. I will get to the title of my presentation about halfway through, but maybe just to get you thinking about this issue of biological complexity. And I don't consider the genome big data anymore, but when I started as a student, actually, about 20 years ago here, it certainly was big data. We just couldn't actually attain it in any way. So just to remind you, your genome is the 23 pairs of chromosomes, which chromosome you get from one of your parents. And there's roughly 30,000 genes strung along those chromosomes that encode proteins. Okay, so these are pretty attainable, manageable numbers. And typically you have 2 copies of each gene. And our big contribution was that, in fact, we found that's not always the case. There's this issue of copy number variation. In some cases you have 1 copy of the gene, in some cases 3. And this becomes relevant to disease studies, as I'll talk about a little bit later. When you look at the gene, so the genome, which is the unit of biology that makes you unique amongst the population, unless you're an identical twin, that's comprised of about 3.2 billion chemical bases of information. So again, that's not that much. When we were involved in Craig Venter's genome sequencing project published in 2007, we got that on a CD, a CD-ROM. You can actually carry it around in your bag on your USB key even. But it's really the combinations of those letters and how they express themselves in this complex environment of genes and environment that, again, make you unique and play into those impacts that affect your medical outcome. And I'll talk a lot about this a little bit later. But the genes only comprise about 1.6% of your genome. And that's where we focused almost all of our research on in the last several decades. And this methodology called whole genome sequencing now allows us to get all of the 100% of DNA. And I'll come back to a little bit later. That's where we need to focus our efforts to really decode what evolution has encrypted in our DNA. And when we alter our environment, it often leads to disease. So the brain is the unit of biology, the organ that actually is the most complex. I showed the slide on sick kids where I work, and the cardiologist would argue with me, but there's really no argument. You can't argue with the human brain. Brendan Frey from computer science tells me that there's computers that are more complex than the brain, but in fact, the newest computers, I'm told, are actually being modeled after the brain. So the brain is the most complex organ or entity that we know of. It's comprised of about 100 billion neurons, or a quadrillion, or that's 10 to the 15th neuronal connections at the synapses. This is, again, relevant to my talk, because I'm going to talk about autism spectrum disorder. And the brain, and I got this off of Wiki, so I don't know if it's true or not, but it can process 10 to 50 petaflops of processing power, okay, in a split second, essentially in every calculation that you make. You're processing your entire lifetime of big data you captured in your big brain. Big daddies have really big brains. Couldn't resist. So in fact, to understand what you are and how that plays into your medicine and your personalized medicine, you have to understand the genome. That's the unit that we can understand. You can sequence it now. You can decode the order of letters, and you can do this really quickly now. So on the right-hand side of the slide, we're showing increasing system price output per day in genome sequencing, and that is decreasing as you go along the x-axis. On the right-hand side are the newest machines that we just got in our lab with sick kids, so the first in Canada. We're running them now. We can do a genome sequence in about 2 days for roughly $1,100 US per genome. So you hear a lot about this. Part of the McLaughlin Center University with Illumina, we're doing a project where we'll be doing 10,000 genomes, ranging from the Rogers Heart Center to cancer coming out of the Ontario Institute of Cancer Research, and I'll talk about our autism project, and lots of stuff in between. But this is amazing when you think about this. Absolutely amazing. We're getting 1.8 terabytes of data off the machines every day. When we bought the machines, we were all worried about the cost of the machine. It's about $1 million US a pop per machine. We have 5 of these now running. We have to put another 3 million actually into the connections to get the data off the machine into the high performance computing facility. No one told us about that. So in fact, generating the data was talked about earlier. It's easy, and human nature, you tend to take the easy route. It's easy to generate data, so people do it. And I really enjoyed the last talk because it's the type of data you look at which is important. So you have to be able to ask the questions, what data do I need to extract? The good data, the bad data, the data necessary to the question to get answers to the solutions for, in this case, for medicine. So data generation is easy, although accuracy of data and validation, all that is really, really important, but you can, with enough investment, you can get to that point. Okay, so, our laboratory, my academic laboratory studies autism, and this is just my introductory slide that ties together my whole talk. So autism, as I'll tell you in a moment, is been defined as a behavioral disorder entirely for 50 years of research. And we've seen stories come up of refrigerator parents or mothers who didn't raise their kids properly, the vaccination hypothesis. My point is that the truth will be found in science, and our major advances have come through big data analysis, as I'll show you. We think that better understanding the different forms of autism, it's really the autism spectrum disorders. It's a collection of well over 100 different disorders that have common clinical endpoints. By understanding each of the different disorders, and an individual will benefit their management. These new genetic tools we have will enable early detection. Okay, so if you can detect autism very early, you can use behavioral intervention protocols and actually help the kids learn a lot of the things they have deficiencies in learning. And the learning problems they have, as I'll show you, is really because their brain is not wired properly. So perhaps they can't process big data in the same way we do, or they over-process it, depending on really the form of autism that they have. Again, as I'll show you in a moment, ASD, there's no medicines for the core features involved in autism, but identifying new targets through genetics allow us to think about new medicines. And perhaps most importantly, I think, and big data is going to really play into this, is because autism, the features affected by autism are the same features that make us most uniquely human, that of cognition, speech and language, how we communicate and how we interact with each other, social behavior. I think understanding these concepts will allow us to understand the brain, how it works, and why we are uniquely human. So this is what autism is. This is, as a scientist, a PhD trained scientist, a genome scientist, I see everything as Venn diagrams. But individuals who have autism are brought to clinical attention. There's lots of us, probably a few in the room, who are Asperger's, very high functioning, who have aspects of social communication deficits. But it's when it goes too far and you can't interact and you have to be seen by someone. Language deficits, stereotypic repetitive behaviors, and there's many different forms of autism, Asperger's syndrome, the Rain Man syndrome, to severely autistic kids that are the stereotypic kids that are typically institutionalized. But what most people don't know is autism comes along with a lot of other medical complications, including intellectual disability. We see 50% of cases, 60% of kids have severe seizures, anxiety, sleep disorders, gastrointestinal problems. So this is a medical syndrome. And as I said earlier, we already know that there's over 100 different disorders that have different names, clinicians' names, that have an autism component to them. So the concept really is that we have to use a personalized medicine approach, understand their genome, so we can best advise the families of what they should do for their children or for that individual going forward. It's also complicated by the fact that because it's a human behavioral condition, you can't diagnose it at day one after birth, because kids don't start to talk until they're 2 or 3, depending if you're male or female. Girls talk earlier than boys do. And they don't interact a lot until they get a little bit older. So the average age of diagnosis in Toronto is actually 4.5 years old. But for optimal behavioral intervention, you need to start even before the onset of the symptoms. As I mentioned, there's no effective core medicine for autism. There's many medical comorbidities. And families will go through this odyssey of diagnosis. They know there's something wrong with their child or something, they have challenges, but they can't get a diagnosis. And this child here shown in this article went through 8 different clinics at sick kids and through Toronto until we ran a clinical microarray, which is small data now, and found that he carried a chromosome 16 deletion, which was really important because it explained to the family they didn't do anything wrong. It was a sporadic change in his genome. It deletes 17 genes on chromosome 16, P11.2, and that was sufficient to cause autism in this individual. In fact, this is the change we see most often now in Canadian families and also in Canadian individuals around the world, but it's a very rare change. It's in less than 1% of the population of autistics. The other interesting finding here is that when we got more data of other children who had chromosome 16 changes, we found that they also had severe anxiety and often were obese. And in some of the cases, it was the drug that they were taking for their obesity, for the anxiety, that actually triggered the obesity. So having evidence of what the genetic cause was of the ASD also gave data on which drugs they should take or shouldn't take. This is what we call pharmacogenetics or genomics. Okay, so this is the evolution of our contributions to autism, going from in 2007, what we thought was big data. We were running microarrays that had 1 million features on a microscope slide, essentially. That was big data back then. And we found that 5% of the children who were autistic, who had no known cause, were carrying deletions or duplications, so-called copy number variants, in critical genes and encode proteins that work at the synapse. It kind of brings together my first slide. Then we got a new array that went up to about, this was 10,000 features. This was 10,000. This is 1 million features in 2010. Found a lot more copy number variants. It accounts for about 10% of the kids. And it gets to my point, in 2013, we published the first paper using whole genome sequencing, so decoding all 3 billion chemical letters. And in this case, it was 33 families. It was a pilot study. We found explanations for the autism, or one of the medical comorbidities I mentioned earlier, in 40% of the families. So getting more of the data, the big data, gave us a lot more answers for the families to start to come up with a plan for the management of their child. And to cut a long story short, this is from a review we wrote recently. There's now over 124 different ASD risk genes identified, and 55 of these C and B regions along chromosome where multiple genes are involved, not just a single gene. And every single one of these has already been shown to be involved in disorders that have intellectual disability. It's really interesting. Okay? Okay, so perhaps that's a major clinical factor, which we, as I said earlier, we know 50% of these kids have challenges with intellectual disability. No single gene or chromosome region accounts for more than 1% of autism. And actually, we published a paper just last year that shows roughly 10% of the kids have 2 or more mutations that lead to autism and something else, or perhaps both lead to a more severe form of autism. So this concept of understanding the genome is really critical because it helps to subcategorize not only autism but other disorders. And in many cases we find other mutations they carry that predict later onset disorders like breast cancer, for example, or colon cancer, and that's a whole area of topic maybe we'll come to in the discussion. So this is the top 6 list of mutations we found in the Canadian population. We follow a cohort of about 3,000 Canadians. We have pretty deep longitudinal data. The chromosome 16 deletions I talked about, the nerexin gene encodes a protein involved at the synapse, a gene on the X chromosome. So I didn't mention that we see autism in a sex bias of 4:1. So these X chromosome genes, if females have 2 X chromosomes, start to explain the gender bias, and lots of others. Now what's really interesting here, and perhaps you know, if I was a PhD student, this is what I would be studying. We have people, actually I saw Curiel's name mentioned earlier, and he's an MD PhD student studying this. You see the same mutation in schizophrenia, can lead to schizophrenia, bipolar, ADHD, intellectual disability, depending on the genetic change. But this is the one we see when you have the deletion of 16p. More often than not, it's autism, but in some cases, a duplication of 3 copies leads to schizophrenia. So it's these imbalances, changes in the homeostasis of the proteins that control the balance of the synaptic proteins lead to this phenotype that we call autism. This is just one family where we can show a deletion in the synaptic scaffolding 1 gene on chromosome 19. Segregates in a 4-generation family. And but if there's any budding geneticists here, what do you see that's different? The answer is actually on the slide too. The black boxes in the pedigree are those that are affected with autism if they carry the deletion. So the female carriers are protected for some reason. This is not on the X chromosome, it's on autosome, it's chromosome 19. So we think hormonal biology and the expression of genes at certain times plays into the explanation here. But anyways, having this type of data at the time, this is actually retrospectively, but having this data at the time would have allowed early diagnosis in these children. In fact, even prenatal diagnosis or carrier screening. And we can do that now for roughly 20% of the families. We have sufficiently highly penetrant genes where we can use these for early detection. Okay, so this is the biology. So we went in 10 years from knowing really nothing, black box, defining some of the genes, then looking, this is the synaptic scaffolding 2 gene. I mentioned 1 before, and there's a 3rd, which is also an autism gene. And what you see here is these genes make proteins that work at the synapse. And at the synapse, you have these spines at the end of the neurons that are involved in communicating with this neuronal interaction, this massive number of petabytes of interactions in the brain. And in those that carry mutations, either a copy number decrease or a gain, you see a decrease or an increased density of the spine volume. You can't really see it so well here, but it's essentially less branches on the tree, if you will, in this case. And that's the underlying biological challenge that these kids have in their brain. So while it is a behavioral disorder now, for many of them, we can actually define it as a genetic disorder and have an explanation. And if you take all of those different genes, and this is big data now, because now we're taking every gene product from, we've looked at 10,000 or so families and looked at their mutations. These are the rare genetic mutations. And using software that was developed by Gary Bader here in the Department of Computer Science at U of T, and some of the work we do together, actually can plot out the interaction of all of those gene products in lots of different experimental systems. And show here that more often than not, the genes product are interacting, the genes products we find in autistic individuals versus controls are interacting with each other. And we can now identify pathways, including the neuronal development axon pathway up at the top here, and a new pathway called chromatin modification. This is involved in epigenetics, how the environment talks to DNA. and then also MAPK signaling pathways. So these are new targets now for new medicines. So we didn't have this data even a few years ago. Okay, so we've kind of cracked open this black box and have some new targets to develop new medicines. So, so a few years back, I mentioned in 2013 we did this pilot project where we sequenced the genomes of 32 families, and that was a multi-million, I think it was about a $6 million experiment at the time. It was really expensive, and someone else paid for it. But we decided that as the cost came down into the $2,500 range this last year, as I said, now we're at $1,100 US, we were going to launch this massive project. So we were going to sequence 10,000 families, their entire genomes, and try to find all the genetic factors involved in autism so we could chart a personalized medical plan for these families. We've done 7,000, and by the end of the year we'll have 10,000 done. Unbelievable. It was just 2007 where we worked on Craig Venter's single genome. So the data is coming fast and furious. So the data will continue to come. We need to think about how to make sense of the data. That's important. And the trainees who are here, the data will come. Think about making sense of it and getting it to your patients in this case. So anyways, not just the genome data, in fact the phenotyping data we heard in the last talk, all the different types of data is very, very complex. So we're developing new tools to capture all that data. Consent forms for research ethics protocols is even more complex. Every jurisdiction has their own rules and interests and cultural attributes, so we have to generate that as a resource too. We partnered with Google because now we have to move beyond traditional high-performance computing facilities and go into the cloud to actually manage the storage of the data, but also the analysis of the data. So we're into the petabytes of information that we've generated in the genome sequence, which we cannot manage on our own high-performance computing infrastructure, even though we have a great one at Sick Kids here at U of T. So the idea then is to put the data in the cloud and make it available, so it's been consented, so that anybody in the world, any student from any place can take their questions, their curiosities into the cloud and perform their own experiments. And to come back to the question, I really enjoyed the discussion at the end, is the key, is to have a background in computer science and medicine, whatever it may be, so you're enlightened and you can think about what that appropriate question should be. Right? And that's going to lead to new discoveries that change things. So I won't go into this detail, but what my laboratory does most is develops a lot of the tools to make sense of genome sequence data. And it's based on a deep understanding of how human genetics works. So we look at all the different kind of features using traditional genetic questions, a lot of statistics we'll hear about in the next talk. And then we compare to all these massive databases that have been generated from groups like at the CCBR Donnelly Center, who are doing systems biology, animal models from around the world, and then come up with a list of genetic variants. It's essentially an Excel spreadsheet. We go from big data to an Excel spreadsheet, try to put it into a form now for doctors using their PDA devices, and Mike Brudno will talk about that later, so that they can actually make a decision on how to chart a course for the medical management of that individual. So when we did this pilot study, we did 32 families. Prior to doing the whole genome sequencing, we would have answers for about 10% of them. We did the whole genome sequencing. These 16 families, we had to communicate genetic information back to them to either explain issues of genetic recurrence risk or drugs the kids should or should not be taking. In some cases, it enabled actually earlier intervention for their siblings. Okay, so we have a new study we're just submitting to New England Journal where in about 5% of kids that carry these highly penetrant mutations that I showed on my top 6 list, if the second sibling also inherits that change, in every single case they end up somewhere on the spectrum. So we can start early intervention really at day 1. And I won't spend, well, I'll talk about this very quickly. This was an interesting latest study where we looked at families that had 2 or more affected kids, and it plays into this issue of genetic heterogeneity. We found when we had information, could find the mutation that was involved in the family, in 50% of the cases that siblings carried different mutations leading to their autism. Okay, so the reason for this is there's a lot of targets, right? And the new mutation rate in the genome is sufficiently high that you see this issue of genetic heterogeneity. It's very similar to cancer. So just quickly, this is a gene that's involved in epilepsy, the SCN2A gene. and it was identified because the technology of whole genome sequencing allowed us to scan exon 18, which is missed by all the other previous technologies. This is another family where we found a de novo, or a spontaneous mutation in another epilepsy gene that is found in the female index case, but the sibling brother inherited a different genetic change in the UBE3A gene, which is involved in Angelman syndrome, which is another form of autism. About 40% of kids that have Angelman syndrome exhibit autism also. This is inherited from, passed from mom to boys. It's an imprinted gene, and that's typically what you see when you have mutations, is it only expresses itself in the male carriers. And this is a great example on the right where we found, again, two different mutations. One, a spontaneous change in the thyroid hormone receptor alpha gene on the left, and there's actually clinical trials ongoing now in Cambridge, and the families we found with mutations are now participating in these trials. And on the right, it's an inherited gene in yet another epilepsy gene. The mutations that are specific, it seems, to these epilepsy genes, if it affects a specific part of the protein, it leads to a more complex autism presentation versus a simpler epileptic presentation. So getting into the big data now, we're generating ideas from the genomic big data that lead us to think about epidemiology, which is really where the big data lies, the longitudinal data, going out and looking at all of the environmental influences, which is everything else other than the genome, really. And this is our first reductionist kind of look at that data, which is pretty interesting, though. You think about the number of the de novo mutations, so the new mutations that occur in the germline are predominantly coming from males, from the fathers, so mutations in their sperm, and that rate of mutation increases with age. That's what these two plots show here. So, in fact, the greatest risk factor for autism right now is the paternal age of conception. Okay. But what we found, this is new data, no one's seen this yet, it's actually in review in Nature right now. If you look at these copy number variants, they induce an aberration in how DNA replicates itself. We found lots of new mutations at the breakpoints of these copy number variants. But for some reason, it only occurs in females. So in fact, the mom and dad are playing into the biology. So here, prior to this new data, moms and everybody's pointing the blame to the dads now. It's no longer the refrigerator mother, it's dad's age of sperm. Which plays into, and this is really, when you think about the theory behind this, is people are having their children later in life now because you require an education. You have to go to school a lot longer. So the MD, PhD students here, you don't start your own lab till you're 36 now. That's the average age. So you have your kids later. So in fact, it's not a genetic issue, even though it's the environment. It's the environment, the social environment that causes you to have your kids later, mutation rates go higher, and it leads to an environmental behavioral condition. So it depends on how you look at things. Okay, so how you frame the question. And then this is work we've done with Ann Bassett here now, where we're looking for, again, quote unquote, we call them mutations because it leads to some type of a measurable clinical outcome. Ann Bassett's group, and we're looking at pediatrics and kids, and Ann's looking at adults who have mental health disorders, in particular schizophrenia or bipolar, and she's finding the same mutations in these individuals. And if we have the connectivity of the longitudinal data in between, like that 4-generation family I showed you earlier, in almost every case when the data exists, we could see hints of these different symptoms, either in the kids or the adults, depending on which way you come at the question. So getting one of these copy number variants in autism, say on chromosome 16, or in this case, chromosome 2q13, predicts that they may have even more complex neuropsychiatric outcomes when they get older. And this is really the essence of how you use genetic medicine or genomic medicine to predict. And it's, we've given the simplest examples, the low-hanging fruit, where you have these highly penetrant, highly impact mutations. And as we get more and more data to compare against millions of genomes, I think we'll be able to really decode the genetic code that has evolved through natural selection and evolution. And this is, I think, my second last slide. This is the called Garrett's outlier. This is the clue to, I think, a lot of the conundrums that I presented to you. This is an amazing family where we see this genetic resilience. So if you look here, this boy came to Sick Kids, and he was autistic, and we ran the microarrays. In fact, we've done the whole genome sequencing now on the whole family. and found that he not only carries the Nerexin-1 copy number variant, that was number 2 on my top 6 list. He also has the MBD5 deletion copy number variant, which I think is number 8 on my list. And he's autistic. His sister has both of these, we found. At the time she was borderline autism, she's still kind of subclinical. She has a lot of features, but she's doing pretty well. But what that means is these changes must have been inherited, right? So of course we went back and looked, and Peter Zatmari, who's here at the university, this is actually his family, and checked with Dad very, very carefully. Dad's just doing fine, he's doing really, really quite well. So he's resilient for some reason, so this is the clue. Maybe it's something he's eating, maybe it's something in his upbringing, maybe he's got other genes in his genome that produce an aberration or a difference in that protein pathway figure I showed you that allows him to escape the more severe attributes of autism. So that's when we get lots of these millions of genomes, where we're going to take this type of approach to not only mapping the genes involved in autism proper, but those that are actually leading to a resilience. And I think this is where the new medicines are going to come. There's lots of projects doing this. And then my last slide is to come back to what I said at the beginning. We've done a really good job studying the 2% of the DNA, which encodes the genes. But in fact, the future is really to understand the 98% that's there for a reason. Sidney Brenner, when he came here to get the Gardner Award a few years back, had this great comment. Actually, I was a student at the time, I remember this, and he said, you know, people refer to this DNA as junk DNA. Evolution, you know, evolution keeps this for some, you know, it sticks around, it's in our genome. And people throw away garbage, but they don't throw away their junk because it means something to them. And that's exactly what's happening here. We just don't know what this 98% of the DNA means. We do know it's involved in turning genes on and off. We just don't know which combinations of the letters. And once we sequence enough genomes in the population, as I said, we'll be able to work backwards and decode how evolution played out. And just by comparative biology, through high throughput data analysis, figure this out. Brendan Frey's group and the team here at U of T is developing machine learning approaches to do what they call deep genomics, and he started a company on this. So he's working with our center.